Hello, this is your introductory lesson for Biology 101 for the first day of class. And what we're going to talk about is sort of these shared characteristics of living things. Biology is the study of life, and so things that are living share certain characteristics. Most of you guys know that living things are built of cells. We're going to be doing a lot with cells in Bio 101. We need to understand how cells work on a cellular level in order to understand how the whole organism works. And so, yeah, living things are built of cells. We have single-celled organisms, we have colonial organisms, which would be two single cells independent of one another that are attached. You'll be able to see some of those in lab, like Anabina. And then we have multi-celled organisms like humans. Another characteristic of life is that all of these cells use DNA in order to reproduce. So we would draw DNA molecules in these cells. Now, prokaryotic cells are a little more primitive. They've been around for a very long time on Earth. Prokaryotic cells. And they do have DNA, but it is not enveloped in a nucleus. Okay, so there's basically means there's no membrane around the DNA. And that in part is because of their life strategy in order to reproduce, in order to evolve through reproduction, they rely on mutation. And having an envelope around your DNA protects your DNA, and that's just not a life strategy that prokaryotes have used. Eukaryotes, on the other hand, do. Humans are eukaryotic, and our cells have membrane around the DNA and all sorts of other membrane as well, membrane-bound organelles, which we're going to talk about in class. Membrane-bound organelles that are used for energy or that are used to produce proteins. So eukaryotes don't rely as much on mutation. We protect our DNA, and instead we do sexual reproduction. This isn't for all eukaryotic cells, but this is for humans. And this would be asexual reproduction for prokaryotic cells, okay? And these guys rely heavily on mutation. So we'll be able to talk a lot more about mutation as the course goes on. All right, the next one would be develop. So that simply means that they develop and become more organized and mature over time. Capture energy from their environments. The food system we're going to study, the food web, is going to be a photoautotrophic food web. So if we think about organisms being able to capture energy, the one that we're going to focus on is the one that uses sunlight to fuel the entire food chain. And of course, sunlight is what plants use along with carbon dioxide and water to go through photosynthesis, and that fuels, of course, the whole food chain where animals are eating plants and animals are eating animals. But just so you know, there's another food web as well that's chemoautotrophic that doesn't rely on sunlight. So there's this whole other world that's happening on Earth where it doesn't use sunlight and chemicals are used instead, just for a little fun knowledge. So if you're thinking about deep sea hydrothermal vents, those are chemoautotrophic food webs. So there are some fun places on Earth where you can see these dynamics. We are going to focus on this photosynthetic dynamic. Sense the environment and respond to it. That's something that even a primitive prokaryotic cell can do. Sensing chemicals, being able to learn from certain chemicals, and evolve even things like behaviors in response to chemicals. We're going to talk about antibiotic resistance in class, and we'll talk about some prokaryotic adaptations to that. And of course, eukaryotic cells can do this too. High level of organization, absolutely. And one of the things that you're going to notice between a prokaryotic cell and a eukaryotic cell is that this eukaryotic cell, the one with the membrane here, it is going to have really, really complex features inside. These membrane-bound organelles are very sophisticated, and you'll see that high level of organization. And then the last one would be to evolve, and evolution is simply change over time. We're going to talk about different ways this happens later in the course, but of course this evolution is using the DNA molecule. So we have all these organisms on Earth, and some of them are more similar to one another than others. And one of the ways that we can classify organisms is by understanding the sequences inside of the organism's genome. So when you read about DNA in the book, you'll read about nucleotides being stacked together, and those nucleotides having nitrogenous bases that are either A, T, G, or C, and those are in a particular order, and that really matters in coding regions that we're going to be able to call genes. Okay, so in order to be living, you need to meet all of these requirements, and if you don't, then you're not living. Now that seems really, really straightforward and black and white, but I do want to show you an example of a gray area. So here is something, we'll just say, that is not necessarily classified as living or non-living. It just doesn't fit well, and these are viruses. And oftentimes these viruses are the ones that make you sick, right? You breathe them in through your nose, and they go ahead and get in through cells in the nose, and they're able to replicate and divide once inside your cells, and can make you sick and cause an immune response. So here you can see 3 different types of viruses, and if you go back to your characteristics for living things, well, they meet some of those, but not all of those. Let's go back. And so what you'll note is that while they can reproduce, some of them only do it using their RNA inside of their protein. Some of them do have DNA. They are organized. They definitely can evolve, some of them more frequently than others. Some only have RNA and no true cells. So you can see here that there really is no true cell. In this picture, they look kind of odd. They kind of look like little spaceships, and that's because they are essentially nucleic acids that are surrounded by protein. So the question is, are they living or non-living? And there's not really a good answer to this. These can be called infectious agents. One of the characteristics about them in terms of reproduction is that they can't reproduce on their own. So what they do is they go into your cells, insert their DNA, and cause your cells to divide. And when that happens, they are able to divide. So that's definitely a different way of thinking about reproduction, and not really sure how that fits into the living or non-living category. Regardless, they're here on Earth, and we are going to be able to talk about them a little bit this quarter. Okay, so it might seem straightforward even after showing you the viral example, but what about some of these other things? When you really get down to it, how do you know what's living and what's not living? Some of these are obvious, so mushrooms are living, the sun is non-living, but some of them are in between. So what about something like pollen, or a single oak leaf that's fallen to the ground? Or what about blood? Are those living? Those are actually not living, but once alive, because those cells do actually have the ability to live for a short period of time. So pollen, blood, a single oak leaf, but not indefinitely, so they will eventually die. So non-living but once alive would be the answer for that. The virus is on there, and that just has a big question mark. Other examples here, something you might not be aware of, is something like a seed. A seed is actually living, but just has a really, really low metabolic rate. Fingernail clippings, that's an interesting one. This actually fits into non-living but once alive. Basically, those fingernails are compressed cells of keratin, which is a protein. And so all those compressed cells make the hard part that you're seeing as the fingernail, and they were once living cells. Okay. So a couple other things before we stop. One is the scientific method. You're going to be looking at two types of research in a lecture as well as in lab. We're going to be talking about experimental versus natural experiments. And in your first and second labs, you're going to be talking about this. So I thought I'd give you a little bit of background here. So for experimental studies, these are highly controlled studies where you actually are using a control group. And when you have a control group in your study, the power of your results goes up significantly, and you're able to go ahead and imply cause and effect. Now, so you can use a scientific method to go ahead and pursue experimental studies with a control group looking at cause and effect. What you guys are going to be doing in lab, the second week of lab, is you're going to be doing a natural experiment where you take a baseline, so let's say resting heart rate, and then you go ahead and do some sort of activity, let's say drink caffeine, and then you go ahead and test your own heart rates again. Everybody participates, and you're measuring against a baseline. That's a natural experiment. It's not as powerful as something that's experimental with a control group. And you're only going to be able to talk about correlational connections. So you could say that caffeine is associated or correlated with a higher heart rate, but it didn't necessarily cause it. And so that's something to keep in mind as you're going through those labs. You're going to be looking at figures a lot in this course, so make sure you understand this is the y-axis. This is the x-axis, and what you have is your dependent variable is always on your y-axis. It's what the results of your measurements, essentially. So in this case, it's corn yield. Your independent variable is on the x-axis, and you can see here amount of nitrogen fertilizer applied was varied. So that was your variable that was varying, and which is the cause, right? Okay.